All right. So, hi, everyone. We are going to go ahead now and do our review over chemistry. This will be a set of video questions like we would have covered in class this week. Of course, we didn't have lecture part of class, so we're just trying to get caught up and stay on schedule as much as we can. So, the reason why we're doing chemistry is because everything in biology ultimately is based on chemical processes. And then from there, chemistry is based on physics, basically. So, we have to have kind of understanding of this to be able to understand a lot of the stuff that we're going to see throughout the rest of the semester. So, going all the way back to the beginning, right? So, atoms are the fundamental units of matter of the universe. They are what make up the elements. Of course, elements are things that cannot be broken down into simpler things using normal chemical means. And as an atom is going to have a nucleus with protons with a positive charge, and normally neutrons with a neutral charge, kind of in the center of the atom. So protons and usually neutrons are in the nucleus, and then electrons are going to be outside the nucleus in what are called orbitals or energy levels, moving really, really fast around the nucleus because the negatively charged electrons are attracted to the positively charged nucleus because of those protons that are there. As far as the kinds of elements that are most important to life on Earth, you're looking at carbon, hydrogen, oxygen, and nitrogen. Those four are going to make up the vast majority of everything in your body as well as any other living thing. Now, a little bit more, involved in atoms are what are known as isotopes. And so an isotope is the same kind of element, but one that has a different number of neutrons than other types of that same element. So, for example, most of the carbon in the universe is what we call carbon 12, and carbon 12 has 6 neutrons in its nucleus. However, there are at least two other forms of carbon. There's carbon 13 and carbon 14. Carbon 13 has 7 neutrons, carbon 14 has 8 neutrons, but they're still carbon. They just have slightly different properties. Okay. And a lot of times those isotopes, especially the really rare ones, are rare because they are radioactive. Isotopes, not super important to living things. There are contexts where they do become important, but for the most part, we don't really worry too much about isotopes in biology. Ions, on the other hand, are a different story, right? So ions are what occur when an atom either gives up or accepts an electron. All right. So one thing that we have to really, really remember here is that we can never change the number of protons in an element or in an atom without changing the identity of that element. So for example, again, if you have nitrogen, which is number 5 on the periodic table. Nitrogen has 5 protons. If you were to give another proton to nitrogen, it would actually become carbon, which is a very, very different thing. Neutrons, on the other hand, electrons on the other hand, you can have different numbers of those in different various forms of the elements, but you just can't change proton number. You can change electron number, but you cannot change proton number. Okay. So these ions are going to become really important to us as we work through the semester. They are going to allow us to explain a lot of biological processes that we are going to see. Moving on to number 3, the atomic mass of an element is, well, actually, let's do atomic number first. The atomic number is the number of protons and electrons that you find in an atom. Okay. The atomic number is how the elements are arranged on the periodic table. They're arranged sequentially from the lowest atomic number, 1 proton, all the way up to the highest atomic number, somewhere over 100, 110, somewhere in there. Those are arranged by the number of protons in the nuclei of those atoms. And because the atoms on the periodic table are balanced, if you know the atomic number, not only do you know the number of protons, but you also know the number of electrons, which can be kind of handy. Okay. What you don't know from that, just the atomic number, however, is the number of neutrons. And sometimes that can be important too. So the atomic mass is going to help us figure out how many neutrons are in the nucleus. Okay. So the atomic mass is the sum total of all the protons and all the neutrons in an atom, all of them. The atomic number is just the number of protons. So if you take the atomic mass and subtract the atomic number, what you get is the number of neutrons in that particular atom. Okay. The atomic mass minus the atomic number gives you the number of neutrons. Okay. Now, the protons and neutrons in the atoms are important, right? They are what give the atoms their identity. They are what determine who the different atoms are, who they belong to, what elements they are. But the electrons are really what we care about in biology. The electrons are going to carry energy with them. And energy is something that all living things need to have, right? So think about electrons spinning around outside the nucleus of an atom. And the electrons aren't completely by themselves. They're also carrying with them a bit of energy. And the amount of energy they can carry can change. They can pick up some energy and they can drop off some energy. Okay. So those electrons are really what we care about most in the biological context, especially when we start thinking about things like photosynthesis and cellular respiration, where we have electrons gaining and losing energy over and over and over again. That's a really key aspect of the atom is those electrons. Okay, now some of you guys, I'm sure, have heard the term orbitals before. I used the term before when I was talking about where we find electrons relative to the nucleus of an atom outside the nucleus, right? Orbitals are like s orbitals and p orbitals and d orbitals and those kinds of things, right? We're not going to worry about that. What we're going to think about in this class are energy levels. Okay, and these two terms are related. The energy levels of orbitals, they are very much related, but we are going to focus our attention on the energy levels. Okay, the energy levels around a nucleus, there can be as many as you need to accommodate all the electrons that are present in that particular atom. The thing about that is that the first energy level can hold only 2 electrons. It might not have any. It could have 1 or it could have 2, but that's it for the first energy level. Every single energy level past that can hold up to 8 electrons. So as we get to larger and larger atoms, we start adding more energy levels to accommodate the electrons that we need to show, right? So energy levels, first one can hold up to 2 electrons. Every one after that can hold up to 8 electrons, okay? And this is something normally we'd show in class on the board, but we can do this next time in class as well, is sort of drawing what this would look like, what's called a Lewis structure that shows primarily the electrons around the nucleus of an atom. All right, now this next number 6 question. Before we talk specifically about number 6 and answer that question, I want to introduce another term. All right, so we'll just kind of add this here, something called the octet rule in chemistry. So the octet rule says that an atom is most stable when its outermost energy level is completely filled with electrons. And that the thing that's a little bit confusing there is sometimes completely filled could mean empty. I know it's kind of like a paradox there, but having a full energy level and having a completely empty energy level, kind of the same thing. Okay, they're equally stable. But if you have energy levels that are not completely filled, that's where we start getting into the reactiveness or reactivity of some of these different types of elements, right? And so there's basically 2 kinds of elements in this context in the universe. There are those that are very stable, and they are happy the way they are. They don't want to react with anybody, because if they do, they're actually going to become more unstable. That's not good. Universe likes stability. On the other hand, there could be reactive elements that can only achieve stability by reacting with other elements. And so when they do that, they can then satisfy their octet rule. Okay, so the idea here is that outermost energy level completely filled makes you very stable. If it's not completely filled, it's going to make you somewhat reactive. Okay, now, thing is, and this is a little bit counterintuitive, but the closer the atom is to achieving its octet rule, the more reactive it's going to be. Okay, so we're going to run down a couple of different examples here, and it's really hard to be able to not draw this on the board for you, but I'm going to just describe it the best I can, and you'll be able to keep up. It's fine. So we're going to look at sodium first. Sodium is number 11 on the periodic table, which means sodium has 11 protons, and it also has 11 electrons that we would need to show in a Lewis structure. So think about sodium. It's got 11 electrons. We know the first energy level can hold 2. We know the second one can hold 8, so that's 10, right? 2 plus 8 is 10. That means we're going to have 1 electron in the third energy level of a sodium atom. And like I said, the closer they are to achieving their octet rule, the more reactive they're going to be. All right, now sodium, 1 electron in the outermost energy level. Highly reactive because it's very, very close to satisfying the octet rule. It has to do one of 2 things. With 1 electron in the outer shell, it has to either give up 1 electron, which is not really that hard, or find 7 more electrons to fill up that outer shell and achieve the octet rule. Now, which of those is easier, giving up 1 thing or finding 7 things? Giving up 1 thing, of course, and that's what sodium does. It likes to give up 1 electron, and that's going to make it a sodium ion, right? So it's going to have 11 protons still, but only 10 electrons. So it's going to be a sodium 1+ charge ion. On the other hand, if we were to look at carbon, carbon on the periodic table is number 6, which means 2 electrons in the first energy level, 4 in the second level. And that puts carbon in kind of a weird spot because carbon either has to pick up 4 electrons or lose 4 electrons if it's going to ionize to achieve the octet rule. And it turns out that's pretty much impossible. You can't really find 4 or give up 4 electrons. It's too hard. So carbon is actually not super reactive. It's kind of reactive, but not like we talked about with sodium or chlorine or things like that, because it's kind of in the middle of having that outer shell filled. It has to either give up 4 or find 4, and doing that's going to be really hard. So carbon, rather than giving up or accepting electrons completely, is going to enter into what are called covalent bonds, where it's going to share electrons with other things in order to become more stable. We'll talk about that more in a second. All right, number 7. Number 7 is referring to a redox reaction. And redox actually is kind of like 2 words smashed together, like staycation. Redox stands for reduction oxidation, okay? These reactions always go together. You cannot have one unless you also have the other one with it, which is why we can kind of combine the terms into one. A reduction reaction is a reaction where something gains an electron. I know, seems kind of backwards. Reduction gains an electron. But if you think about what kind of charge the electron carries, right? It's carrying a negative charge. So if we stick another negative charge onto something, right? Well, what are we going to do? We're going to reduce its charge. If we add one more negative charge, we are reducing the overall charge of that atom. So that's why it's called a reduction. If you add an electron to something, you are reducing its charge. On the other hand, oxidation is where you take an electron away from something, okay? And it turns out that oxygen is a very, very good oxidizing agent, which is why the name is kind of similar, right? So the idea here is that when you oxidize one thing and take electrons away from it, they have to go somewhere. So if you oxidize one thing, you automatically have to reduce something else. The electrons from the oxidized thing have to go to the reduced thing in order for this all to make sense. Okay. Number 8, ionic bonds are bonds that form due to an attraction of charge. Okay. This is where we're going to have a positively charged thing attracted to a negatively charged thing. The old saying that opposites attract, right? So with sodium, we already talked about that one before. Sodium likes to form an ion with a 1+ charge because it only has 1 electron in the outer shell. And with 1 electron in the outer shell, if it gives it up, it achieves stability and becomes that Na1+ ion. Chlorine, on the other hand, a little bit bigger. Chlorine's number 17 on the periodic table. So I'll just run that real quick for you. That means 2 electrons in the first shell, 8 in the second shell, 7 in the third shell. So that takes us to 17. That means chlorine either needs to find 1 electron or give up 7 electrons. And just like we saw with sodium, the 1 is much easier than the 7. So if sodium gives up 1 electron and becomes a positively charged ion, and chlorine accepts an electron and becomes a negatively charged ion, we have a plus charge, we have a minus charge, they attract. That creates an ionic bond, which is what holds sodium chloride together, among other things. It's an attraction of charge, right? So it turns out ionic bonds are of some importance in biology because they can disrupt various kinds of processes, and they can sometimes be used in various kinds of processes as well. Sodium especially, chlorine, calcium, potassium, those are all really, really important ions that sometimes form into ionic bonds or can sometimes interfere with certain other processes, and not necessarily a bad way either. But ionic bonds are not quite as important to biology as covalent bonds are. Covalent bonds are where electrons are shared between 2 atoms. It's not that one electron is being given up completely and the other one accepts it and takes it forever, or sort of forever. It's more so the electrons are bouncing back and forth between the 2 atoms that are sharing the electrons. Okay. So just like any other relationship where you share something, there's always a possibility that your partner might not share equally with you. And so it turns out that different kinds of elements have a different level of what's called electronegativity. And electronegativity is the affinity for electrons that an atom has or an element has, how much it likes electrons, how strongly it pulls electrons. That's electronegativity. Okay. So if you have something with, say, a really high electronegativity, like oxygen, sharing with somebody else who has a really low electronegativity, like hydrogen, for example, then even though the hydrogen and the oxygen are sharing electrons, they're not sharing them 50-50. The electrons are going to spend, like, we'll say 90% of the time with the oxygen, with a high affinity for electrons, and they're only going to spend 10% of the time with the hydrogen because it has a low affinity for electrons. Okay. Now, what you then have to think about if that happens is that 90% of the time, those electrons are going to be hanging out with oxygen, and oxygen is going to have a slightly negative charge during that time. And when the electrons are hanging out with oxygen, that means they're not hanging out with hydrogen. So that means hydrogen is going to have a partial charge, partial plus charge during that same time. Now, the electrons do go back and forth, so it's not like it's a permanent charge. It's not like sodium chloride. The electrons do bounce back and forth between both the H and the O, but they spend far more time with the O than they do with the H. So that's what we call a polar covalent bond, a situation where two elements are sharing electrons, but they're sharing them unequally. And therefore, there's going to be a difference in partial charge throughout the overall molecule. The entire molecule is still neutral. It's still balanced, but the electrons are hanging out asymmetrically with different parts of that molecule, making those parts slightly negative and making other parts slightly positive. On the other hand, a nonpolar covalent bond is where you have completely equal 50-50 sharing of electrons. Okay. So that is going to occur when things have either similar or identical electronegativity values. Sorry, I was looking for something. Okay. So if they have similar or identical electronegativity values, they are going to share the electrons equally, and there is not going to be any sort of difference in charge throughout the entire molecule. Okay, so the thing about polar and nonpolar molecules is that molecules that are held together by polar covalent bonds are what we're going to call hydrophilic or water soluble, meaning they dissolve in water, they like water, they interact with water, they're cool with it, right? Because water is also polar. Polar things dissolve in polar things. On the other hand, nonpolar substances are going to be hydrophobic, and they are not going to interact with water. Oil is a molecule held together by hydrophobic bonds, nonpolar bonds, and therefore it does not mix with water very well. Okay, so polar dissolves polar, nonpolar dissolves nonpolar, but polar and nonpolar do not get along very well. Why? The reason why oil and water don't mix is because polar and nonpolar things don't really get along very well. I'm going to hold off on hydrogen bonds and show this in class next week, because it's something where I really do need to draw this one to be able to show it very well. So we're going to skip that, and I'll just draw a quick picture on the board next time we meet. In terms of ways to increase the rates of a chemical reaction, basically, you've got a few options, but only one of them really works reasonably well in living things. Okay, so one ability, or one thing you could do to, if you want to make a reaction go faster, is you could heat it up. That's why chemists are always with the Bunsen burners and the hot plates and those things. You got to heat stuff up all the time. Okay, that doesn't work if you're a cell. If you heat up a cell, it dies. And that's going to slow down the rate of reaction at that point. So that's not going to work for a cell. Another option is you could increase the amount of substrates or reactants you have in your reaction. And that could also allow you to create more products as well, do the reaction faster, right? But again, that's not always an option. We can't always just have more substrate laying around in our cells. So that, even though it could work sometimes, probably doesn't most of the time. Third option, if you want to increase the rate of reaction, is to add a catalyst. All right. So catalysts are chemical substances that have the ability to greatly accelerate the rate of reaction. In biology, we call those catalysts enzymes. All right. So enzymes are proteins. And just like any other protein, their recipes are written into the DNA. So if the DNA is altered in some way, then the protein might not work correctly, and therefore the enzyme is not going to function the way that it should. So that kind of takes us back to our DNA protein conversation that we had before in class. If the DNA is flawed in some way, the protein is flawed in some way, and it's not going to work the way that it's supposed to. So that's going to be a pretty big deal, as we'll talk about. As far as how enzymes do what they do, we'll talk about that more in a couple of chapters, kind of beyond the scope of what we're doing here, but something we'll talk about, I think, in chapter 4 or 5, I think. Okay, mentioned before that water is a polar molecule. And so water, therefore, has some special properties that are unique to water, and properties that really allow for life to exist on Earth. Water is sometimes referred to as a universal solvent, which is a bit of an exaggeration because it's not universal. Water will dissolve lots and lots of things as long as they're polar, but it really doesn't do very good at all with non-polar things. All right. So water trying to dissolve oil, not going to work very well. Water trying to dissolve salt or sugar, not a problem. Right. Um, because water is a polar molecule, it has the ability to adhere to other substances that are also polar. We call that adhesion, of course. When you guys saw your meniscus in lab, right? So if you're looking at a graduated cylinder with water in it, it's not a flat line across the top, it's cupped, it forms a meniscus. And that meniscus forms because on the sides of that graduated cylinder, the water is sticking to the glass. In the middle, where it's not near the glass, it's kind of being pulled down by gravity. So you get that meniscus effect. And glass is a polar substance, so water is able to adhere to it. Cohesion is where water molecules are going to stick to each other. So this is the reason why we have surface tension on water, right? So if you were to do a belly flop into a swimming pool full of water, it hurts because you have to break surface tension to enter the water. If you were to do a belly flop into a pool of oil, first off, gross, but you would not have the same degree of surface tension, would not hurt nearly as much because of that lack of surface tension, right? So this is the reason why those little water striders can walk around on the water surface and ponds and lakes and things like that. There's surface tension there. When water changes its physical state, when it goes from water to vapor, that allows for cooling of surfaces. This is the reason why sweat cools off our bodies when we're too warm. We're actually pulling heat away from our bodies in order to evaporate water, and that causes the body to cool down. Water is also a really good temperature buffer, meaning that you have to it can absorb a lot of heat, and it can also give off heat slowly for a long period of time, which, living near the Great Lakes, is kind of the reason why we have slightly warmer temperatures near the lakes than we do kind of away from the lakes. Of course, more extreme if you go near the coast, the ocean, a lot more water there, a lot more kind of a stabilization of temperature. One thing about water that's unique is that solid water is less dense than liquid water. Normally, a solid substance is going to be more dense than the liquid form of that same substance, but water is the opposite. That's a really important thing if you happen to be an aquatic insect that wants to survive winter in a very, very cold climate. Because if it was the other way around, if solid water was more dense, then ice would form on the bottom of a lake, and it would start to form layers all the way to the surface, and everything inside of it would freeze solid. But instead, ice is less dense than liquid water. It forms on the surface and forms a protective, almost like blanket over the water underneath to help keep it from getting any colder and from freezing solid. So that's very important to things that are aquatic. We're not going to worry about the climate thing. pH is something, I think we'll show that in class as well. We'll just take a look at a pH scale and talk about exactly what this means and how we calculate it, why it's important, that kind of a thing. We'll add that to our discussion of hydrogen bonds when we talk about the class. Same thing with buffers. Okay, so the next thing we need to do, talk about some biochemistry. And this is really where we start to get into the importance of chemistry, specifically to living things. So the molecules we're going to be looking at in this next section are all organic. They're all carbon-based, okay? Carbon can form 4 bonds. Because it has 4 electrons in the outer shell, it can share with up to 4 partners to achieve its octet rule and achieve stability. All 4 of these organic molecules are going to be constructed from monomers that are joined together to form polymers. All right, so if you look at the prefixes, mono is 1, poly is many. So a monomer is like a single building block, like a single Lego. And a polymer is many of those building blocks hooked together, like the Lego thing that you built, right? So the 4 categories of organic molecules that we're going to see are carbohydrates, proteins, nucleic acids, and lipids. All right, so we've got those 4 structures. Now, the nice part about all 4 of those kinds of molecules is they all have this monomer-polymer relationship. They all have that going on. They're all formed the same way through what's called dehydration synthesis, and they're all broken down the same way with what's called hydrolysis. All right, so dehydration synthesis means you're going to pull out a molecule of water. That's the dehydration part. If you have 2 of these building blocks together, if you remove a molecule of water from both of them, kind of collectively, that's going to create a bond, which starts to form the polymer. Okay, so you remove the water molecule, you create a bond, building blocks start to get hooked together, and eventually you end up with either a dimer if there's only two of them, or a polymer if there's more than two of them. But we're not going to really talk about dimers too much. Okay, so they're built the same way. They're broken down the same way. Hydrolysis is where you insert a water molecule to break the bond that was formed through dehydration synthesis. So it's like the opposite of that process. Dehydration synthesis builds, hydrolysis breaks down. Okay, so first of those four organic molecules is carbohydrates. Carbohydrates, building blocks, are referred to as monosaccharides, single unit sugars, things like glucose, fructose, galactose. Those are all monosaccharide sugars. Disaccharides are where you have two building blocks hooked together. So that would be like sucrose, which is table sugar, maltose, lactose is another one. Those are all disaccharides. And then polysaccharides are where you have more than two, usually several hundred or several thousand building blocks hooked together. So polysaccharides are things like starch and things like glycogen in animals, chitin in fungi and things like that. Those are all polysaccharides. So carbohydrates really can be used for one of two things in a living system. The more important of the two is energy. All right, carbohydrates are your go-to energy source to build ATP to keep your cells going right this moment, right? That's kind of the main function of carbohydrates in our particular context. The other thing carbohydrates can be used to do is they can be structural, right? So for example, plants, the main body of a plant is made out of a polysaccharide called cellulose, which is not an energy carbohydrate. It's a structural carbohydrate that makes plants have some strength and rigidity to them, okay? We're moving on to proteins then. Proteins have as their building blocks something called amino acids. And there are 20 different amino acids that are used to form every single protein on Earth by putting those amino acids together in different orders and different lengths to create different shaped proteins. All right, so proteins are, of these four organic molecules, the one who is most reliant on its shape to function. Protein shape and folding is its most important feature. If you take that away, the protein doesn't work anymore. Proteins are also the most diverse category of organic molecules. They're the most common type of molecules in a cell. About 80% of the dry weight of your cells is protein. Okay, the rest of it is the nucleic acid, the lipid, and a little bit of carbohydrate left behind. Most of your cells are made of proteins. Uses can be everything from being an enzyme to being a receptor, being a transport protein, being a hormone, almost anything, right? Proteins are extremely diverse in what they can do inside of a cell. Now, one thing we want to go back and think about is, how do our cells know how to make their proteins? Of course, that comes from the DNA, right? The DNA is the instructions that we need to build the proteins. But if you change that DNA, like we talked about before, through a mutation, then the protein is going to come out differently. And there's a really good chance it's not going to function anymore because it's going to come out the wrong shape and therefore have an incorrect function. All right, when that occurs, we have a process called denaturation. And that is the unfolding of a protein that typically leads to that protein losing its functionality. Okay. Things that can cause proteins to denature are things like high temperature, really salty environments, like high salinity environments, environments where pH is especially acidic or especially basic. Proteins are super sensitive to those kinds of things and will begin to denature almost immediately if the conditions are not right. And again, if they unfold themselves, they're not going to work anymore. All right, nucleic acids are next. These are things like DNA, RNA, ATP, and a few others that we'll talk about. These are made out of nucleotides, but for right now, I just want you to know that term. It's the building block for nucleic acid. Don't worry about specifically its composition yet. We'll get to that later on. Nucleotides are held together by what are called phosphodiester bonds that creates, for DNA at least, that double helix that we're probably all fairly familiar with. We're going to see this plenty more in class, so I'm not going to describe it now, but like I said, we're going to get probably tired of hearing about it this semester. ATP is the stuff that keeps our cells running. It's energy. It's the stuff that's the energy source for this particular moment in your cells. ATP energy is keeping your cells alive and allowing them to do everything they need to do. The central dogma of biology. Okay, so a dogma is like a widely accepted set of ideas. And the central dogma says this. It says that DNA is used in the construction of RNA, and then RNA is used in the construction of proteins. Right? That is how life works on Earth. That is the meaning of life on Earth. DNA is used to make RNA. RNA is used to make protein. It almost always has to go in that order. There's one exception. There's a group of viruses called retroviruses that can actually go backwards. They can go from RNA to DNA, but that's a story we'll talk about at the end of the semester. All right, the last one, lipids. Lipids are made up of what are called fatty acids. Lipids are things like oils and fats and waxes. There are hormones that are lipids, insulation, long-term energy storage. Phospholipids make up the plasma membrane of a cell, and that's a really critical component of cell structure is its membrane. So lipids are also pretty diverse. They can also be used for waterproofing, keeping water in to a plant, for example. So pretty diverse in function. Not so diverse in terms of structure as proteins, but still we find a lot of uses for lipids. Saturated, unsaturated trans fats, not super worried about that for this class, so we can go ahead and skip that. And that kind of brings us to the end. So I know I went kind of fast, but there's no one on here and no questions being asked, so that tends to happen. Okay, so we'll see you guys back in class. Hopefully this was useful. Come with questions if you have any, and we'll get caught up on Tuesday.